can't hear her anymore.
Okay, we're live. Hi, is anyone listening to me yet? I don't think so.
Wow. Hello. Who are we talking to?
Right now, no one, but
The
posterity.
internet. Okay.
Okay.
I don't know.
Yeah,
How
so
does
this
that happen?
Yeah, I can do it for you if you want. It's a YouTube, it's called like a YouTube live event. And then anyone who has the website can watch live and then record it. Okay, so.
I want you to be in it.
Calling it.
Jill's sister, a couple of friends, and
Donna, Ed's mom,
My mom?
Lauren and Simon.
my mom.
Yeah, she asked
Oh.
my other aunt.
Jill, are you gonna stand like here?
Yep. Great. Oh, good, that works. That's great. Great.
My parents, one of my brothers, and then
That was
an uncle does consulting in San Diego, so he's sticking around.
Perfect.
Yeah.
Other than that, great. And then just be out now, everyone's feeling much better. Great, good. Sit home, relaxing. This is the office.
Great.
Will we know if they bring your phone? Is it loud enough?
Well, you know what?
If Kelly and Dave bring your phone?
Yeah, you should.
Okay.
We have not called it. Do we have or need water or anything? I have water, thank you.
Super.
Julie, did you take this photo? No, I found it on the internet. She found it herself. I found it. Oh gosh.
What?
I just looked at the screen.
Oh, that one? We can cover the screen.
That's great.
You don't have to
Yeah,
look at
oh,
the
that's
phone.
great.
Yeah.
No, that's Jen.
I'm just gonna make it straight.
Uh,
Is this? Do you like this one or do you need it?
No, it's great. It's great. I just looked at it. Okay. Thanks for helping out with the parking situation. Yeah. No, I was just watching. How much is this? Depends. We're lonely.
Do you have a room? Great.
Yeah.
Right at the certificate.
I've been doing it for the past.
I've got a Bob response to an email as a PDF.
This looks good. The first part,
Yeah.
and then the last third is a little wordy. Because I thought they kept responding the same thing.
Is this capture?
I was really insistent on the
Okay.
whatever it was.
Okay, I'm gonna hop into.
My mother says the picture is okay, but the sound could be better.
Baby, the same one.
My mother says.
Tell
Jill, what if you just pretend to speak?
Hi, Olga. Hang on. Are you gonna be? Where is she? Okay, we're doing another sound test. This is about the volume at which I will be speaking. I can see how my hair looks now, so I just to fix it.
Are you gonna be mic'd?
Nope. Is it better? Can you?
I don't know.
Okay.
I mean, it's up to her to text me back.
Okay, great.
Is your sister online?
I don't know.
She has a darn good reason for not being here, don't you think?
Oh, hey, do you mind running down just to clear some? Let's just stick it in my dashboard. Thanks.
Okay.
Hi, Dr. Vu.
Oh my gosh, there are two differences. You thought about the lighting.
Yes.
And that there's air conditioning.
Oh my God, there's air conditioning in here. It was so hot for yours.
Yeah. I was just saying, oh, it was there.
know, I was like, I'm gonna be sweating.
I'm gonna be
Great, great.
hello.
Yeah. Do you want, yeah, I was like. It's an outfit. Yeah.
Hopefully we'll be needing to gather some signatures.
Hopefully. And I have 4 pens. Just in case.
Just because.
Yep, Jill. Cause like how horrible would that be? Oh God.
Don't worry, I've got this.
Okay.
People often just ask me, so. I was like,
No, we'll do that later, once you have the camera going.
It's about putting these presentations together, I should just make sure that you guys each get a copy of yours, because it's like a.
Yeah. Okay, move on. Good one.
So I'm now just getting, that is like a hybrid costume. So I'm thinking initially, like, oh, it's just some standard student attire.
Yes, my subtle Halloween outfits, we are like professional. It's in the office.
So Jill's sister, by the way, said in here. Yeah, so she's home with, they're saying and watching on her, I can't see it.
Yes. But like nothing's happening right now, right?
That's likely what's going on.
So she.
Yeah, I don't know. Yeah.
So Jenny, years ago, we were in Santa Barbara.
Yeah.
December, you happened to be this grandfather was in Santa Barbara, and she came to meet you first.
Yeah.
And I like dropped her.
What?
Say that one. Oh, we didn't know that. Somehow there was some coincidence that we were all in Santa Barbara at the same time.
Yeah, well, no, I was living at.
Oh, you were living there.
I didn't know. She is like salty now.
See, I took her out, she bought some clothes for that meeting, because we were like, oh, this is a really cool person. She had a clothes.
Yeah.
Similar life. She's pretty excited about it here. So it's funny, at the same college, she's only in the a couple weeks in March. We're happy to host this one.
Did you find them?
Yeah, I gave them the thing, there were no spots in the main lawn at this level. And
Are they going down?
so we told them that,
Perfect.
I don't know.
Can you give me your phone?
But now this is all they gave you. Oh, it's normally like your. We sailed and we had a black night. We were like, wow. Pretty diverse. Climb on tall shoulders.
You need Simon Shoe like
What?
you need Simon Shoe.
Probably right after.
What is that?
Probably all like walk down there.
So many people calling.
Okay. So many people here. Yeah, that's so awesome. It was really neat to meet them through the International Center and kind of know some people.
We have anybody?
Good, thank you.
Oh,
I
I'm
Right.
It was really nice to come.
Thank you. Oh, I need him for all the things that we've agreed to do while you're gone.
Yeah, get there, get there.
Just stop. I mean, I told him that I would process the data, so we just need.
Well, he's now chartering another boat from land.
Okay.
So we'll have 18 right now. That's finally-
This is my face drifting into the corner. Is like when you start a GoPro video underwater and you see like a scuba diving face. I'm like, is it on?
It's the rest of, yeah, I don't understand that. Okay. Yeah. Yeah, I don't think I've done this now, but I would get somebody else to edit it. It's just-
Do you want to stick your face in there? It's recording.
So, nobody's in there?
So it's not like a video conference, it's just a one-way broadcast.
Who's in there?
Two of my aunts, my sister and my baby niece, as mom, Lauren Freeman, Donna Brown, what?
Festivities to come. But we're all here today to celebrate and listen to Jill Harris's PhD defense. I'm Jen Smith. I'm Jill's PhD advisor. I'd like to welcome Jill's family here from out of town, from the DC area, as well as Jill's committee and all of her friends and extended family and colleagues that are here today. So I'm gonna, as with tradition, give a brief introduction into Jill and her history as a scientist. As with my normal kind of preparing these presentations, I always do a little bit of Google stalking to see if I can find any embarrassing, incriminating photos. I was specifically trying to find photos of Jill from when she was a child, and there are no photos of Jill pre-master's thesis, really, so I was pretty convinced that Jill arrived on the planet as a master's student. But thanks to Jill's sister, Jill had a very happy and fun childhood with her parents, Jeff and Joan, her sister Yana, in the DC area. Jill always had a sense of humor, and as with all children, not every day is perfect. Your sister. I had to use them all. And then Jill went on into her more professional side of life and did her bachelor's at Dartmouth and went on to do a master's at the School of Marine and Environmental Affairs at the University of Washington. And as far as I can tell, Jill's passion for the ocean really began when she began as a science and math instructor at the Island School in the Bahamas, where she taught high school students how to do science and math and spent a lot of time in the water and snorkeling and diving around the reefs in the Bahamas. From this point on, Jill was motivated to go on and into a career in marine sciences, and she enrolled as a master's student at the University of Washington, working on the social ecological dynamics of marine protected areas in the remote parts of the Philippines. So she spent a fair amount of time traveling around the Philippines and interviewing local communities about what worked and what didn't work in marine protected area management. I think around that time, somehow Jill found me when I was a postdoc at Santa Barbara, at the University of UCSB. At NC's, and I remember Jill setting up a meeting with me, and it was like, wow, there's a student that wants to do a PhD with me, but I'm not even a professor. So I don't know exactly how Jill found me, but a few years later, I happened to have got lucky and landed a job here, and Jill circled around and joined my lab. So Jill joined the Smith Lab family. Of course, at the time, the lab didn't look like this. It was pretty much just this. Loretta still likes to fall asleep a lot better, as we see in this picture. So as with most of my new students, when they join the lab, we have an active research program on Maui, and so Jill got to join us all over on Maui at the SLIMEO, the Smith Lab interim Smith Lab interim Maui extension office. And Jill was a founding member of SLIMEO. So we went over to Maui, and this is where Jill first began her scientific diving career. Started off a little rocky with fins and arms and hair and legs flailing and scuba hoses and whatnot. But over time, we worked it out and everything was going to be okay. And Jill learned her passion and love for seaweeds, the little slimy fuzzy thing that she's holding in her hand. And that set her off on her career in my lab, studying the ecology of the small things that most people don't notice on reefs. Jill's time in my lab took her all far and broad. She spent time in Brazil, working in the Abrojos Bank, studying the coral reefs of these remote offshore reef locations. She joined us on the 2010 Line Islands cruise, where she was part of the benthic team studying the health of benthic reef ecosystems across a large group of islands. We were aboard the beautiful Hansa Explorer. We were all, everyone on this cruise was all very happy we were not aboard the Moa Moa, which we all saw was this island, which actually was floating and was transporting people around. Jill went to Australia for the International Coral Reef Symposium with our lab family. She, as you can see, we have quite a lot. There, Jill is falling asleep again. She also joined us to Palmyra Atoll, where she conducted some of her PhD research looking at ecological dynamics of turf algae on coral reefs. Jill then went on to the Maldives with her lab mate Levi, where they studied a variety of different things. One piece of work from this is currently published and is part of Jill's PhD thesis. She then went to Curacao. Yes, I'm still going. She conducted a large part of her research there on Curacao, on the island of Curacao in the Caribbean. And so you can basically see that during Jill's time as a PhD student, she's also learned how to get a PhD and how to travel the world on your advisor's time. So this just shows a smattering of the places that Jill's been. During her PhD, she's actually been to Scotland and BC for conferences, so you can see Jill's journey has taken her literally from one end to the other. So we should all take some lessons from Jill if we want to get more traveling in our life. During her time here, she learned how to become a rigorous ecologist, a phycologist, or someone who studies seaweeds. She spent many, many hours behind the microscope. She guarded her samples with her life. She also became a fantastic mentor, where she literally took this one student, Nico Pavlanis, under her wing and mentored him as an undergrad and all the way through to his graduation, where now Jill and Nico and I actually have a paper that's about to get published. So she really embraced this mentoring capacity. Sometimes she wasn't always sure about what she was doing, and she admitted that she needed help, and so this is where the advisor was there's myself too. So we always work things out. Along the way, Jill learned how to have a great time, how to love Mexican food. She probably already did anyway, but you'll notice that over half of the place in the view there are actually Jill's fajitas. She made a lot of great friends along the way. Had a great time both in the field, here at Scripps with fuzzy friends and friends in breweries. She also became an auntie and I guess has become an auntie for the second time and really embraced this, where wishing her nephew Owen happy birthday from underwater on the remote island Pacific. Jill also embraced the holidays that we're about to embark on here, Halloween, taking on a variety of different morphotypes, including dreidels. Jill always took the opportunity to do science education and outreach, whether it be at a conference, wearing 3D glasses, or at a bar, talking to your general public at the events called So Two Scientists Walk Into a Bar. Jill was always happy to talk science, especially if beer was involved, especially if it included craft beer like we have so abundantly here in San Diego. You'll see Jill here embracing the beer socializing again. And sometimes even PBR was okay, but most often it was craft beer. So wherever there was beer, Jill was happy to talk about science. She also learned how to become a team player, playing paintball with her fellow lab mates. She also became a great team member, a team player in our lab family of the Smith and Salmon labs. You can see here that we all love to take group pictures, whether it be at sustainable seafood events, at Christmas, where we can wear sparkly things, or at conferences, or in our backyard at our annual Christmas party. So ultimately, I hope you've seen through this journey that Jill has traveled the world. She's become a critical and analytical and rigorous marine scientist. She's got strong analytical and quantitative skills. She's become a great science communicator and educator. She's become a wonderful mentor, a fantastic team player, team member of our lab, as well as a friend. So I thank you for all of that, Jill. And the fun doesn't end here. Jill actually has a job waiting for her in DC with WWF, where she'll be traveling the world again, going to Indonesia to study the effects of marine protected area establishment in Indonesia, looking at what works and what doesn't and how to make marine protected area establishment more effective in the future. So without further ado, I will invite Jill to tell us about her adventures as a PhD student. We have some traditions. Sorry.
It's a little bit.
Okay. Other people. Okay.
Hang on, we have a spinning pinwheel right now. How's that? Good. Great. Good. Hi, good afternoon, everyone. I'm in a really great mood. I'm so happy to see so many people here. Thank you to my family, my parents, and my grandpa for coming all the way here to see me. Thank you to my committee for your excellent support along the way. Thanks to everyone who's online watching us, including my newest niece, who's just a baby, can't be here, but she's watching. Okay, so today I'm going to be telling you about the ecology of turf algae on coral reefs. I'll start with a really sort of big picture view of why coral reefs and why they're interesting, why they're worth studying. They're incredibly diverse. This is one of my favorite pictures from one of those places around the world that Jen talked about. And one of the reasons it's so beautiful is because of the diversity. There's many, many species we can see in this photograph, and coral reefs are some of the most diverse ecosystems on the entire planet. They're also very important for food and livelihoods. They support, mostly through their fisheries, they support people all over the planet for their food and for subsistence. The physical structure of the coral reef provides really critical shoreline protection in many of the coastal areas of the world. And finally, because they're so beautiful, because of all the fish and the corals, they're really important for tourism. They bring in hundreds of billions of dollars of tourism money each year. So they're incredibly diverse and incredibly important. But I am a benthic ecologist, so this is actually not how I typically look at coral reefs. This is a more accurate perspective of how I look at coral reefs. We're looking at the benthos now, the bottom of the coral reefs. I spent a lot of time during my first couple of years at Scripps looking at photographs like this. So this is the typical way that benthic ecologists study coral reefs. We take a picture of the bottom, we identify all of the organisms in it, and then we calculate the percent of the reef area covered by each of those organisms. So in particular, I was looking at photographs of the reefs from the Central Pacific, these sort of dark orange areas in the middle, which are the Pacific Remote Islands Marine National Monument. And I spent many hours looking at thousands of photographs in those locations. They're highly protected, they're uninhabited, they're some of the most remote, pristine coral reef ecosystems on the planet. And when I was looking at them, I was seeing lots of corals, but I was also seeing lots of turf algae. So that's all the stuff I've highlighted here, and I'll just toggle back and forth. All of the stuff under the green, that's all turf algae. And I'll tell you more about them in a minute, but they're these small species of seaweeds that make up huge proportions of the benthic reef environment. And if we look at the data, we can see that turf algae make up about 30% of these really remote coral reefs. So these are plots of the benthic cover data from those 7 islands in that Pacific Remote Islands Monument. And we're just looking at the green bars at the top part. So there's a little variation from island to island, but in general, turf algae make up 30, even 40% of these coral reefs. So it's not all coral. So I began thinking more about what the turf algae are and what they're doing. They are small. They're small species of seaweeds, usually less than a centimeter tall. What's really important is that they're multi-species assemblages. So there are many, many species that make up these turf algae assemblages, and they're heterogeneous. And I'll talk, the rest of my talk today will be about this sort of diversity and heterogeneity of these turf assemblages. And we like to call them sort of a shag carpet on the coral reef. They're these fuzzy, multicolored carpets of algae. They're ecologically important for several reasons. First of all, they're incredibly productive. This is a photograph of turf algae with little oxygen bubbles trapped inside because it was producing so much oxygen. The oxygen was coming out of solution and actually getting trapped in the turf matrix. They're incredibly important food for herbivores. Basically, every single herbivorous fish and urchin that lives on a coral reef primarily eats turf algae for its main food source. And finally, they're really important competitors with corals. So corals are the organism that actually builds the coral reef. Everyone's competing for space on the benthos, and turf algae are the most common competitors with corals. In addition to being ecologically important, I also predict that they will become more abundant in the future. And that's because turf algae are what we call pioneer species. So they're some of the first groups of organisms to settle on the reef after new space has opened up following a disturbance. So here's this litany of assaults, anthropogenic assaults that are facing coral reefs today and degrading coral reefs. And all of these things that are bad for corals are actually good for turf algae. So here's a photograph on the left, and the middle photograph are photos of bleached corals that have recently died from bleaching events. This is sort of a bird's eye view of a big table coral, and that greenish thing, the fan-shaped thing in the middle, is turf algae that's just taken over and has moved out and is covering that formerly live coral. The one in the middle is from the most recent bleaching event that's still going on, I think, in Maui. And if you were to look closely at this picture, you would see that that coral is actually still alive. There are little live polyps in there, but it's being overgrown so quickly with turf algae that it probably is not going to be able to recover and survive. And finally, on the right is a photo of a coral covered with being killed by disease. That sort of reddish brown band is a disease front moving across the coral. And right behind that disease front is that greenish yellow turf algae coming in behind it and taking over. So the traditional way that we study turf algae misses some of the important details that I'm going to be talking about today. So as an analogy, we can look at this forest canopy photograph on the left. We can look at that forest canopy and say, okay, that's 95% tree. And that would be correct, it's about 95% tree. But we know, we sort of have this intuitive understanding that we'd be missing a lot of the detail if that's how we described it. We might want to know, for example, what species of trees are there? How tall are the trees? Are all of the species clumped together in one space, or are they spread out? Are there animals that live in the forest, and how are they interacting with each other and with the trees? And now the traditional way we study turf algae is sort of that same way. We would look at that picture on the right. This is a picture of a mostly dead reef, and we could look at that and say it's 95% turf. And that would be accurate, but we'd be missing the same amount of detail that we're missing if you describe the forest that way. So for example, let's say there's an endangered species in the forest that we care about, and it eats fruits from a particular tree. We might want to know, is that tree there, and is it spread out, and is that endangered species going to be able to survive? Similarly, in the turf analogy, there are specific species of turf algae that are particularly detrimental to baby corals that are trying to settle. So we might want to know, are those species of turf algae there? Are they distributed evenly? What can we predict about the future settlement of coral on that reef? So I'll give you some close-up views of turf. Here's a photograph of turf competing with coral. So on the left we have live coral. On the right we have a really thick, dense turf community that's growing over that coral. This one has a pretty tall canopy height. It probably sticks up a couple of centimeters. It's really dense. You can't see the underlying substrate, and it's packed really completely with sediment and sand and detritus. Now contrast that with this photograph of turf algae. So here's a little baby coral that's been settling, and all of this fuzzy stuff around that coral, all of those filaments sticking up, those are also turf algae. But it looks really different from the previous photo, right? It's much shorter, there's no sediment packed in there, and I would look at this and say, well, that baby coral is probably going to be fine. There are probably a lot of herbivores eating that turf algae and keeping it really cropped. Now here's a third example of turf algae. We're looking down again, bird's eye view, or scuba diver eye view. There's a live coral on the top of the screen, and all of this stuff down here is turf algae growing over a previously live coral skeleton, so over the dead coral substrate. And this turf algae also looks really different. It's really sparse. You can see these bright green splotches. You can see pink splotches. You can also see some of these white marks in here. And those are areas where fish have actually eaten so much of the turf that they've grazed down to the underlying substrate. Now, these three photographs are really different. They look really different. You can see some of the heterogeneity in turf. But the traditional perspective would just lump all of those things together as turf algae. And what I'm looking at now is what are the differences among those communities and what can that tell us about the reef, broader reef ecology. Now, if we bring this back into the lab and look under a microscope, we might see things like this. These are some of my favorite species of turf, some of the most beautiful ones that we see under the microscope. These two are not microscope pictures, but everything else is. And this is just to give you an idea of some of the things that we see when we take a closer look at turf. So my talk today, this is the outline. We'll talk about why turf algae. I've gone over this a little bit, and I'll go into a little bit more depth, including a framework of a better way to study and quantify turfs. My second chapter will look at spatial patterns in turf algae. So there's this central tenet to ecology that we first study pattern, and then we can understand process. So we describe the pattern in an environment, and then we look at potential processes that might be causing those patterns. In the second chapter, we'll just look at the pattern side. And then in chapter 3, we'll take a look at some of those processes that might be controlling the patterns we see in chapter 2, and we'll focus on two processes known to be important on coral reefs, which is consumption and resource availability. And finally, in the last chapter, we'll focus a little bit more specifically on one process, looking at the small invertebrates that actually live inside the turf matrix and see how they're interacting with the turf algae. Okay, so let's start with chapter 1. I wanted to start with a quantitative analysis of how much turf is there on coral reefs, because it's not uncommon to read in the literature sentences like, turf algae are the most abundant benthic cover, or turf algae are becoming more abundant, but there's no quantitative data to back that up. No one's ever done a global analysis, a global quantitative analysis of how much turf covers coral reefs. We do have meta-analyses of coral cover on coral reefs. So here's two from the last decade. On the left is a graph of coral cover on the Great Barrier Reef over time. On the right is a figure of coral cover across the Indo-Pacific. And this is a really good start. We need these quantitative meta-analyses. But if you look at the y-axis of those two plots, they don't go up to 100%. So what is the rest of the reef, right? It's not a black hole, there's something there. And these meta-analyses miss a lot of the other benthic components. So we know there's coral. We also know that these meta-analyses are missing macroalgae, turf algae, of course, and crustose coralline algae, or CCA. And in fact, many monitoring programs lump some of these groups together. They'll lump, for example, all of those, the macroalgae, the turf algae, the crustose coralline algae together with sand, with bare space. And these groups are really ecologically different, so we wanted to focus just on turf algae and get at some of this variability. So we collected data from 8 regional monitoring programs. Originally, I started looking at 29 programs, but I had to eliminate all but 8 of them because most of them just didn't record data on turf algae, or they lumped it together. You'll see that the data are not orthogonal, so I don't have every single country, evenly spread across the world from every single year. We have a pretty good spatial coverage. We're a little bit data sparse in the Indian Ocean, but we're pretty good for the Pacific and the Caribbean. And we think that this is our estimates are probably conservative estimates because we don't have this, perfect spatial coverage. So we ended up with this global meta-analysis of 176 records from 35 countries over the last 20 years. And this is where a record is an average for one country or one island in 1 year. So the answer to the question, how much turf algae is there, is 39.7%. And what this means is that on average, coral reefs around the world, 39.7% of coral reef area is covered with turf algae. And this is pretty close to the figure I showed earlier that showed that 30% figure across the Pacific remote islands, right? There's a little bit of variability. These are all the different countries from which we have data. There's a little variability from country to country. But in general, globally, we're looking at 20 to 60% of reef area covered in turf algae. Now these are island-wide or country-wide averages. And if we look at the site scales, of course, there's greater variability at the site or reef scale. Some sites have less than 1% turf algae. Some sites have greater than 90% turf algae. But this is a pretty good estimate. So what does that mean, 39.7%? Let's go back to this picture at the beginning. This was from a remote island. It is inhabited, but it's sparsely inhabited. This looks pretty healthy. But this coral reef probably has 30 to 40% turf algae. So that gives you an idea of what that figure looks like. So I started off the talk saying that if we just describe turf algae with this broad brushstroke of spatial coverage, we're missing that detail, right? And then that's exactly what I did. So to remedy that, I proposed a new framework for studying turf algae that gets at some of that variability based on 4 turf algae metrics, each of which address some or all of those 3 important ecological characteristics that I spoke about earlier: production, herbivory, and competition with corals. So canopy height tells us simply how far above the substrate, how far above the coral does the turf algae go. It's equivalent to, tree height or canopy height in a forest. This is a pretty thick, lush turf community that goes up to a couple of centimeters. Most turfs are much, much smaller on the millimeter or sub-centimeter scale. Sediment content is how much sediment, sand, or detritus is trapped within the turf. This photograph looks like there's sand stuck to the side of that coral. It's actually turf algae growing over the coral, but there's so much sediment in that turf community that we can only see the sand. Density is a sort of a proxy for how much grazing pressure there is. So here's a view of a huge dead coral. Of a huge dead coral covered in turf algae, and all of these sort of lightish, whitish blue areas are bite marks, again, from fish. So this area has really, really high grazing, and all of those bite marks are open substrate in which new turf could settle, or perhaps a baby coral could settle. So density gives us an estimate of that. And finally, community deals with the species composition and the type of taxa that we might see within that assemblage. Now, in particular, I want to talk about canopy height and why that's important. Canopy height determines what is called the boundary layer thickness. And the boundary layer is basically how fast is the water flowing over the surface of the rugose surface of the turf algae. So this is our rough turf algae surface down here, and then these blue arrows are the free stream velocity, just the regular water flow that has no friction, that's flowing above the turf. And if you get right down next to the turf algae, those water molecules right next to the turf are sort of sticky. There's friction. They're not moving at all because they're getting stuck to the turf. A little bit higher up, there's a little bit less friction. They're moving a little bit faster, et cetera, et cetera. So the boundary layer is this area, I like to think of it as sort of stagnant or sticky water that gets stuck to the turf algae. If the turf is taller, higher canopy height, that boundary layer will be thicker. If it's shorter or scraped down to the substrate, the boundary layer will be lower. And the boundary layer doesn't, the molecules within that boundary layer don't diffuse into the overlying water. So you might, for example, get a buildup of microbes within that boundary layer, and they would respire, and then you would get low oxygen conditions or extra viruses, or sort of different water conditions than you would get in the free-flowing water above. So we're going to talk more about canopy height later, and boundary layer is one of the reasons it's so important. Okay, so chapter 1 summary. I've showed you that turf algae are ecologically important, and I've made this prediction that turf algae will become more abundant in the future. I showed you that turf algae make up, on average, 39.7% of coral reefs worldwide. And finally, I talked about some of the multiple characteristics of the heterogeneous turf community and how that can tell us about broader reef ecology. Another way to summarize chapter 1 is in haiku form, which is, what are turf algae? Productive and abundant on reefs everywhere. Okay, so we'll move on to chapter 2, where we will look at some of the spatial patterns in those heterogeneous turf algae metrics. So to get into spatial patterns, I designed a hierarchical sampling scheme at 4 spatial scales. We went to an atoll on the Maldives, and we visited 4 islands. At each of those islands, we visited 4 sites. At each site, we sampled from 4 different colonies, dead coral colonies covered in turf. And at each colony, we collected 4 samples or 4 punches of turf algae, separated by only a couple of centimeters. To do this sort of sampling, I had excellent help from Levi, a great field assistant helping me out with this project. And we had these predictions that there would be different processes affecting the turf at each of these different scales. In particular, we are interested in the role of herbivory, but we know that there's other physical and biological processes that affect turf algae. So what we found is that the smallest spatial scale explained the most variation. And I'll walk you through this plot. So these are hypothetical results. This is a hypothetical NMDS plot, which is a non-metric multidimensional scaling plot. Each of these dots is one turf algae sample based on the community composition, the different species and their relative abundances. So 2 dots that are closer together are more similar than 2 dots that are farther apart. So we thought we might see this type of plot, where you can see those 4 islands grouping out really neatly, and then within each of those islands, you have those 4 sites. Within each site, you have 4 colonies, etc. And now if this were the case, we would see this sort of pattern. So along the x-axis is increasing spatial scale, from the centimeter scale up to the island scale, and along the y-axis is the percent variation explained by each spatial scale. These are our actual results. This multicolored shotgun blast. We did not see that there was a lot of variation at the island scale, and in fact, our data are the exact opposite of those hypothetical results, where we saw that there was the most variation explained at the smallest spatial scale. So what this means is that two patches of turf algae that are a couple of centimeters apart are no more similar than two patches of turf algae on two different islands separated by 10 or 20 kilometers. So there's a lot of variability at the small spatial scale. We also looked at different multiple metrics. When we looked at density of turf algae, we saw the exact same pattern where percent variation explained was the greatest at the smallest scale. We saw the same pattern with canopy height, and we saw the same pattern with species richness, how many species were there. So why does this happen? Why do we see small-scale patchiness characterizing turf algae? And this is our model, this is our prediction for what's happening. So we know from other branches of ecology that spatial heterogeneity is driven by disturbance frequency and disturbance intensity. And on coral reefs in particular, we know that herbivory is one of the most important forces that structures coral reef algae. It's a little bit of a preview for my next couple of chapters, but you can take my word for it for now. So we think that what's happening is that there's small-scale herbivory that's creating this mosaic of herbivore bites. So imagine you have this blank white space, it's open space on the reef. Turf can grow. Remember I said it's a pioneer species, and so it settles quickly. Turf can grow into this light green turf community. From there, it can be grazed back to empty space, or it can continue to grow to a turf community that is dark green. It has a taller canopy height, is more dense, etc. And from there, it can also be grazed back to the beginning. So let's say we start with a totally homogeneous patch of turf algae, and then a fish comes along and takes some bites, but there's other pieces of turf that do not get grazed, and so they become that dark green turf. And then this happens again, and some new spots get grazed and some old spots grow, et cetera, et cetera. So you would end up with this small-scale mosaic, this small-scale patchiness. And the centimeter scale is about the size of a fish bite, so this sort of works on that small scale. Now, this could be repeated over many islands, and if you had roughly the same herbivore population act in the same way on many different islands, you would see the same type of small-scale patchiness repeated at larger scales. So this is our model for why we think we see that small-scale patchiness. In summary, we've learned that turf algae are heterogeneous. We take a quantitative look at that, and they're particularly so at small scales. And we can also look at this in haiku form, which is turfs all look the same, but are they homogeneous, or does scale matter? And now we know that they are not homogeneous and scale does matter. Okay, so we'll move on to chapter 3. So we saw the spatial patterns in chapter 2, and now we'll look in particular at these two processes that control those spatial patterns. So the title of this chapter is Context-Dependent Effects of Consumer versus Resource Control. And I'll walk you through that title. This first part, second part, consumer versus resource control, is also called top-down and bottom-up control. So let's say we have some thing of interest. It can be controlled, the abundance of it can be controlled by consumption or by resource availability. This is perfectly analogous to the economic concepts of supply and demand, controlling the availability of your kale salad or whatever you're interested in. And on a coral reef, when we talk about this, we're talking about herbivory, it's top-down control. Turf algae, of course, is our thing of interest. And then nutrients are our resource, or bottom-up control. And this has important implications for conservation and management because pollution is affecting the amount of nutrient that we find on reefs, and overfishing is affecting the amount of herbivores that we find on coral reefs. Now this idea has been really hotly debated in the literature. Many people think it's only bottom-up or it's only top-down control, the other one doesn't matter. I think part of this debate comes from the fact that this is a context-dependent process. So imagine for a moment that we have these two different coral reefs. On the left, we have a reef where there are many consumers. These are all herbivores. They're all eating turf algae. And the water is really clear. It's really beautiful. So let's just say that there's not a lot of nutrients on this reef. Now, the picture on the right, there are very, very few consumers. In fact, the only consumer in this picture is Levi. He's more of an omnivore than a herbivore. And the water is a little bit cloudy. And so let's say that there's a lot of nutrients in this water. And so that's many resources. So these are two really different contexts, right? Now let's say we want to do an experiment to see if top-down or bottom-up control was more important. If we put a cage on the reef on the left to keep out fish, we're changing the environment quite a bit. There's a lot of fish and we're keeping them out. So we might expect there to be a huge impact of putting that cage down. Whereas in the reef on the right, there's no fish to start with. So if we put a cage down, we're not really changing the conditions. So we might expect not to see an effect of fish removal in the situation on the right. So to test this idea, we use turf algae, which are a very useful tool for this kind of experiment. First of all, because we have multiple response variables we can measure. Many other experiments that are similar to this use macroalgae, where they only look at, for example, biomass or extension length. So we have all these different response variables we can measure to look at different ways that the turf community responds. And second, I showed you that turf algae are everywhere, so it's really nice to be able to replicate the experiment on reefs around the world. To do this, we have this sort of experimental setup. We have a plate of turf algae. It's a piece of dead coral covered in turf. We put it into this experiment where we attach it inside and outside the cage to keep out fish. We attach a little slow-release nutrient ball so that fertilizer or nutrients can slowly dissolve into the environment and wash over the turf. And we end up with these 4 treatments. So we have with and without nutrients, inside and outside cages. Now I said we replicated this all over the world. We went to these 3 different locations that were each different in their environmental context. So Curacao has pretty low herbivore biomass and pretty low nutrient levels. Palmyra has pretty high herbivore biomass and sort of moderate but still relatively low nutrient levels. And the Millennium has high herbivore biomass and really, really high nutrients for a coral reef. In order to replicate this type of experiment, you need a lot of help from people. So I had really excellent colleagues who were co-authors on this paper, Jen, Lindsay, Emily, and Maggie, helped with the experimental analysis and deployment here. They were wonderful dive buddies. So the answer we found is that consumers are a stronger influence than resources. And I'll walk you through this plot. So here's a plot of canopy height of the turf algae across these 3 different locations. We replicated the experiment in Palmyra on 2 different years. And those 4 treatments are along the x-axis. This is a plot only of canopy height. We did look at those other metrics, but I'll only show you canopy height today. The other vectors had pretty similar patterns. Now the first thing to point out is that there was no effect of nutrients. So the nutrients are the circle, with and without nutrients are the triangles and the circles. And within each of these color groups, there's no significant difference between treatments that had nutrients and treatments that did not. We did, though, find a strong effect of caging, strong effect of removing herbivores. So these blue bars are the turf canopy heights outside cages where herbivores were present, and the red bars are inside cages where fish were excluded. So we saw generally taller canopy height inside cages with no herbivores. We can also look at this in picture form. These are photos of the actual turf plates that we used in the experiment. The 2 on the left are the plates outside the cages, and the 2 on the right are the plates inside the cages. You can see the plates on the right just have more turf algae. They look darker, they're fuzzier, it looks like there's more stuff growing on the plate. On the left, I want to point out, if you can sort of see these vertical red stripes running down the middle, the technical term for that is turf mohawk. And that's the area that was underneath the zip tie where we attached the turf plate to the experiment. And the zip tie acted sort of as a de facto fish exclusion device. The little fish mouths couldn't get under the zip tie, and that's basically the only place that turf algae could grow outside cages. Now I said that we wanted to compare this response among locations. So to do this, we calculated an effect size, the response ratio. So along the x-axis are those 4 different locations, and as we move from Millennium to Palmyra to Curacao, we have fewer and fewer herbivores. And then the C is for the cage treatment where we removed herbivores, the N is for the nutrient treatment, and C times N is the interaction term between the 2 treatments. For those interaction terms, if we're at the 0 line, we're seeing simply an additive effect. So having 2 treatments, there's no interaction, we're just adding them. If we're above the line, there's some sort of super additive or synergistic effect, and if we're below the line, there's an antagonistic effect. So we found that there's a little bit of difference among these locations. We see not super strong responses, except when we were looking at Curacao. What I do want to point out is this pattern in the response to herbivores, where we see an increasingly strong positive response to herbivores as you get lower herbivory, as you move along the x-axis. And this is, we argue that this shows that a loss of herbivory leads to positive feedback, creates a positive feedback loop. And I'll explain what that means. And I want to say first that this is context dependent. We see different responses in these different environmental contexts. So this sort of helps resolve some of the debate in the literature. Now this positive feedback, we think, is operating under a similar mechanism from what I described for the previous chapter. So let's say we have, remember we have this bare space, turf can grow or it can get grazed back. What I didn't tell you earlier is that these 2 processes of turf growth are different. The first stage, from bare space to new turf,
is basically recruitment. A new filament, a new algae filament has to settle and grow. And that's a slower process than from light green turf to dark green turf, which is just expansion or growth. So existing turf filaments that maybe have been cropped by herbivores can grow. They don't have to reproduce and settle as new filaments. So what we think is happening is that on Millennium, there are so many herbivores and the turf is so grazed that that when you lose herbivores, the first step is a slower step to respond. And in contrast, on Curacao, you basically have no open space. We're starting with medium to light green to dark green turf, so that when you remove herbivores, the turf is primed and ready to respond more strongly. Now, the other thing we found was really cool was that consumers decrease and nutrients increase diversity. And I'll walk through what we mean by that. So this is a prediction from a 2002 paper that looked at the context dependence of top-down, bottom-up control, consumer versus nutrient control on plant diversity. And this is based on data from aquatic and terrestrial and marine ecosystems, but they didn't have any data on turf algae. So, but many other ecosystems. So they predicted that you have this sort of hump-shaped relationship between nutrients and plant diversity, such that you get peak diversity at mid-levels of nutrients. They predicted the same sort of hump-shaped relationship between plant diversity and consumer pressure, or grazers, such that you get peak diversity at medium grazing pressure. And those combine into this context dependence. So let's say we're over here at low nutrient levels. If we're at this low grazing and we increase grazing, we're going to decrease plant diversity. If we're over here at high nutrient levels, if we go from low to medium to high grazing, we're going to increase diversity. So we looked at this with our data, and we found that, okay, yeah, on Curacao, nutrients increase diversity. So remember, Curacao had the low nutrient levels and the low herbivore levels. We kept grazing the same, and we enriched the turf, we added nutrients, and we saw an increase in plant diversity, turf diversity. On Millennium, we saw that consumers decreased diversity. So that's this blue arrow. Remember, Millennium had the highest nutrient levels and pretty high grazing pressure. We kept nutrients the same, but we decreased grazing, we put the turf inside a cage, and that caused a decrease in turf diversity. So one explanation is that, okay, yeah, great, this is great, our data fit this model. But the only way I could get those red and blue arrows to fit with this model was to put them on the very, very edges of the plot, on the very extreme edges of the plot. So the other possibility, which I think is a little bit more exciting, is that turf algae just don't fit this model. That I think it's pretty unlikely that I just happened to choose two coral reefs that were at the very, very extreme end of the possible coral reef spectrum. I think the more exciting possibility is that we're seeing sort of new ecological processes in coral reef, in turf algae that don't match what you can see from other ecosystems. And that is a really exciting possibility. So in summary, for chapter 3, we're seeing that herbivores control the amount of turf algae on reefs, and that a loss of herbivory can lead to a loss of resilience. Second, we saw that turf assemblages might not follow all of the rules in other ecosystems. And our haiku for this chapter is nutrient supply versus herbivore demand, which limits the turfs. And now, of course, we can see that mostly it's herbivore demand that limits the turfs. So we'll move on to our final chapter. In chapter 3, we saw that mostly it was consumers that drove the amount of turf algae on a reef, but we only looked at fish as consumers. So now we're going to take a look at these small invertebrates that actually live within the turf community. And it's pretty standard to just consider fish as the only significant grazers on coral reef turf algae, when in fact there are many, many abundant and diverse small invertebrates that live inside the turf that use it as a habitat. Now here's a picture that gives you an idea of what that might look like, or a super close-up view of some turf algae from Brazil. There's a little fish staring at us in the middle of the screen here, and then this little blue spindly thing back here is actually a marine spider, which is really cool, and that's what I was trying to take a picture of, but the fish got in the way. So this is, the fish is maybe 3 or 4 centimeters long. The spider is maybe a centimeter or 2 in diameter. But imagine that you were even smaller. Imagine you were just a millimeter or 2 long. This would be this amazing heterogeneous tropical forest that you could live in. It provides a really nice complex habitat that can protect you from predators. So this is what these small invertebrates are seeing as their home. Now my question was, what are these small invertebrates doing? Most of the research to date has focused on just the pattern side of things, quantifying how many invertebrates there are, what species there are. I wanted to start looking at the process and figure out what they're doing. And I developed these 3 hypotheses based on what small invertebrates do in other ecosystems, because no one's really looked at their role in turf, coral reef turf yet. So most of these hypotheses are based on the role of small invertebrates in seagrass beds, in kelp forests, and in freshwater streams. So my first idea was that invertebrates could be eating turf, right? They could be consuming the turf algae or the epiphytic microalgae that grows on the surface of the turf and blocks some of the sunlight for photosynthesis. Second, they could be eaten by fish. So that's a possibility. This would make them really important trophic links in the food web between the small detritus in the turf and these larger predators. Finally, I thought that they could be supplying nutrients to turfs via excretion. So one hypothesis we had for why nutrient addition didn't have an impact on the turf algae is because of that boundary layer. We thought that maybe the boundary layer is just preventing any nutrient in the overlying water from physically reaching the turf algae. So if you have small invertebrates that physically live within the turf matrix, they might be supplying nutrients directly to the turf. Now these would have various positive and negative impacts. So if invertebrates were eating turf algae, that would be negative for turf, that would be bad for turf. If they were eating the epiphytic microalgae, that would be positive for turf. If invertebrates were being eaten by fish, maybe that has no impact on the turf whatsoever. Maybe it has a negative impact on turf if omnivores are eating turf algae along with the invertebrates. Maybe it has a positive impact on turf if the omnivores are essentially getting distracted and eating the invertebrates instead of the turf. And finally, we assumed that supplying nutrients to turf is good for the turf algae. So to test this idea, or to test which of these mechanisms might be happening, we did a really similar experiment to the previous chapter. This is Lindsay deploying the experiment on the reef in Curacao. So we had turf plates inside and outside cages. We added nutrients, and we also added an insecticide treatment, so that slow-release insecticide and slow-release fertilizer could wash through the turf algae, removing invertebrates and adding nutrients. So we ended up here with 8 different treatments. Outside cages, we had no nutrients, yes nutrients, and then with and without insecticide, that gave us all grazers had access, only fish grazers had access, and the nutrient addition to both of those treatments. Inside the cages, again, no nutrients, yes nutrients, and then we had invertebrate grazers only with nutrients, and no grazers at all, both fish and invertebrates excluded. So what did we find? I'll walk you through this plot again. So this is similar to the previous plot we saw, but now this is the change in canopy height over the duration of the experiment. So if we're at this 0 line, that means basically that turf growth is about equal to turf consumption. There's no net change. On the left side of the plot are the samples inside the cages. On the right side of the plot are the samples outside. Sorry, left side is outside the cages, right side is inside the cages where fish were excluded. And we have those 8 treatments along the x-axis. So the first thing we found is that once again, nutrients had no impact. So okay, good, this confirms the previous chapter. Again, nutrient treatments are circles and triangles. And within each of these boxes, there was no significant difference of adding nutrients. What we did find, though, is that there was a very significant difference of removing invertebrates. So outside the cage, these are basically our control treatments. There's no net change in turf canopy height. That's great. Turf is sort of static. The ambient turf growth equals the ambient turf consumption. That makes sense. But now when we added insecticide, when we removed invertebrates, we saw a decrease in turf growth. This means that invertebrates were having a positive effect on turf algae, right? Because we removed them and turf did worse. Now when we went inside the cages, everything's above that 0 line. So we exclude fish, the turf algae grows more. That makes sense with what we saw in the previous chapter. But now adding insecticide inside the cages, removing invertebrates, that positive effect disappears. There's no effect of removing invertebrates if you're already inside a cage. So what's going on? No effect of invertebrates when fish are excluded, a positive effect when fish are present. And here's our model for what we think is happening. So the invertebrates are simply using the turf as habitat. There's no positive or negative impact when there's no fish around. When there are fish present, the invertebrores are eating the invertebrates. And then when we exclude the invertebrates, those fish shift their behavior somehow to herbivory and start consuming more of the turf algae. So that these invertebrates are sort of like a smoke screen. They're being eaten instead of the turf being eaten. And this actually matches up with some of the data that's out there. If you look at the stomach contents of many herbivorous fish, We call them herbivores, but they're actually eating a lot of invertebrates as well. So in summary, for chapter 4, we've seen that this is some of the first data showing that small invertebrates have a positive impact on turf algae. And there's some unknown interaction between invertebrates and fish, and this is going to be a really cool mechanism to test in the future. And of course, I have a haiku for you. This is my favorite of all 4 of them. On tiny algae and tiny invertebrates, small things matter too. So the overall summary of my dissertation is that I've shown you that turf algae are ecologically important, they are abundant, and they will become more abundant on coral reefs in the future. In particular, I showed you that turf algae are heterogeneous, and it's that small-scale heterogeneity that really characterizes turfs. We've seen that herbivores control the amount of turf algae on reefs, and a loss of herbivory can potentially lead to a loss of resilience. This one's really exciting. We saw that turf algae might not follow all the rules of other ecosystems. And finally, we saw some evidence that small invertebrates interact with fish to have a positive impact on turf algae on coral reefs. So I want to leave you with this picture that we saw at the very beginning. Remember this relatively healthy coral reef. There's a lot of coral on the benthos here. There's also a lot of turf algae. I think if you're going to walk away from this talk today knowing one thing, I want you to know that turfs are abundant. They're incredibly abundant. They deserve more detailed study because they're heterogeneous and there's interesting ecological processes going on within the turf and between the turfs and the broader reef community. And I think that in the future, if we want to be able to understand coral reef ecology better and understand and protect coral reefs in the future, we really have to understand turf algae ecology. This is maybe, I have a little bit of stars in my eyes for my dissertation, but I really think that coral reef ecology really needs more turf algae ecology. And with that, I have a lot of people to thank. My committee, it was always a little intimidating to walk into a room with all 5 of you, but I think just the right amount of intimidating, and I really appreciate your support and your guidance along the way. The grad office here is just wonderful in supporting students and getting us to this day. I have many funding sources, some of whom are here in the audience today, and it's just so lovely to have both financial support and moral support and have you guys here listening to me talk. Oh, I forgot to show this picture. So this is, Jen claims to be a phycologist, but she's pretty good at catching fish too. I feel really lucky to be a part of the CNBC community, led by Lisa and Dick, and it's just a really wonderful intellectual community to be a part of here at Scripps, the Center for Marine Biodiversity and Conservation. I did this work at many, many field stations all over the world, and the colleagues I met at those stations and the staff, really just a wonderful group of people to work with. I have some informal and formal collaborators at SDSU who are co-authors in some of my papers and who have been with me in the field for some of these projects, and they're just really wonderful. My lab family is just totally awesome, and I just couldn't ask for a better group of friends and colleagues. My broader SIO family have gotten me through the, right. Yeah, tough times. My actual family is supportive in more ways than I can ever say, and thanks for flying across the country and from Alaska to be here. And I will leave you with this picture of these wonderful smiling faces. And with that, I am happy to answer any questions you have. Yes.
I love picturing the balls that you have,
Mhm.
and the water, but I wonder if you track the water currents at all to see if maybe there might be a certain direction water currents are flowing, or it might go to the side of the cage.
Yeah, we did not track the water. So the question was about how the slow release nutrient balls actually delivered, like, were they successful in delivering nutrients to the experiment? We didn't track current direction. The cages were sort of haphazardly placed all over the reef. It wasn't like they were all lined up in a row. And the site that we did that, in Curacao, the site we did the experiment on had currents flowing in both directions depending on the tide. We did measure the nutrient levels in the water. And for one of the experiments, they're beautiful and perfect and exactly what you would expect. And for the other experiments, they are not beautiful and perfect. So this is where we got this idea that maybe the nutrients are doing something weird in the water and not physically making it to the turf algae. Yes.
Great talk.
Did you like my discussion of boundary layers and physical biological interactions? Thank you.
So, speaking of that,
Yes.
actually, it's slightly different, I was wondering whether you see that, but, so don't take this as, insult, but you're young, and
Thank you.
when I was young, and went to Olympic coral reefs, they looked, my vague sort of impression was maybe they were more coralicious than they were in their pictures. And I'm wondering about long-term trends, and 20, 30 years ago, was the abundance of turf algae as high? Were they as important? Are there going to be turf reefs in
Yeah.
20 years?
Yeah, good and sad question about the long-term trends of turf algae and coral cover on coral reefs. One of the reasons I wanted to do this meta-analysis is to answer that question. Most people will agree with what you said, which is that coral reefs used to have more coral and less turf, but there's no quantitative data on that. The amount of turf algae, there's always been a lot of turf algae on reefs, and some of those early studies from the 50s where they went to remote islands in the Pacific before we blew them up. There was a lot of turf algae on those reefs even then, but I think if we went back today, there would be more. So my prediction is that the amount of turf has been increasing over time, and I wanted to sort of get this meta-analysis out there now so we can collect better data in the future and get future trends.
And so my follow-up question is what you were asking me to ask you, which is, now that we're going toward more turf algae, is the rugosity of the reef changing, and is that affecting things like algal settlement or nutrient transport or things like that?
Yeah, absolutely. We know that big fish like big holes on reefs, and so rugosity is good. Turfs don't erode coral reef, but they also don't build it up. So corals are going to build up and give you that rugosity, and other invertebrates will bio-erode and make it less rugose and sort of more fragile. So turfs aren't going to affect things in either direction, except insofar as they prevent one of those processes from happening. On a small scale, there's a small scale question about coral settlement and larval settlement.
Yeah, just the fact that if you lose a lot of that large scale structure,
Yeah.
you're going to change the turbulence of the reef. And so
Yeah.
they're more granular sort of boundary layer.
Yeah, I would expect so. If you sort of have like a smooth, fuzzy surface covering everything, yeah, I think that's probably a side effect that's going to happen. And one of the pleas I make in my first chapter is for people to start measuring canopy height, because it does affect boundary layers, and it would be cool to have more data, sort of like reef scale data from different experiments and how it affects those processes. Yes.
For the one who likes the coralicious look,
Yes.
is it fair to consider that turf algae is a weed?
What is a weed?
An undesirable organism that's competing for the same nutrient space.
Yeah, is it fair to consider turfs as a weed? Generally, yes. So, I'm a scientist, I'm not supposed to be biased, but I also like more coral. But what's really important is that because turfs encompass, from the really short cropped things to the really dense, thick mats packed with sediment, not all turfs are bad. So if you have turf algae that's finely cropped, that has a lot of open space, that's fine. Corals can settle, and it's not going to affect. It's sort of a common part of coral reef health, coral reefs. The problem comes when you have few herbivores in an environment and lots of sedimentation, and everything gets trapped in the turf, and then it grows more, and then no fish are eating it because there's lots of sand, and it sort of takes over. I think so. Yeah, turfs are weedy, and depending on the type of turf, they're good or bad.
Well, I'm just saying because at least if I understood, part of the premise was we're going to see an increase in the turf algae while we see simultaneously an increase in coral.
Yeah.
So that changes the whole ecosystem unless something is done to modify
Yeah.
the growth of the turf algae.
And if you lose coral, if you have a reef that's 95% turf, like why are we even calling it a coral reef, right? Yeah. So, I don't want to lose my job security, but it would be nice to have less turf on most reefs. Yeah.
In your second chapter, you emphasize the importance of small scales based on seeing the amount of variation at that small scale. And you sort of connected that to the idea of fish bites, possibly. And I wonder if you, what is your reason for emphasizing fish as opposed to maybe small invertebrates also varying at that small scale and them providing mechanism, or just saying that there's a certain amount of randomness in the scale?
Yeah, I think the randomness is probably also happening. So what I didn't get into a lot here, but we talk about in that paper, is that turfs have many, many different growth forms. So you get some that are just filaments that grow straight up, some that are crusts that sort of like fan out over the bottom, some that are like creeping filaments where they send out a runner and then little upright filaments. So just because of that diversity, you're gonna have small scale variability, right? If you happen to sample on a crust that's all of one type versus many, many filaments. And so I think part of that small scale heterogeneity was just the diversity of the sort of morphologies. There is a paper by Stewart that looked at the spatial heterogeneity in different types of grazers. So if you have a fish grazer that's swimming around and biting here and biting there, that creates a really different pattern from having an urchin that like shuffles along and sort of munches as it goes. And so I think there could be some other types of grazers that are affecting the heterogeneity. That centimeter scale patchiness just like matches really nicely with fish bites. So it was a pretty parsimonious answer, but those other processes are probably happening as well. Good question. Yeah.
Given that you're moving into more of a marine protected area management associated job, and given what you know about turf algae and their potential increase on reefs globally, how would you go about trying to remedy some of these things, or what would you recommend, or what might you do as you move into more of a managerial position?
So Jen's question was about how to remedy this proliferation of turf algae that we see. I think that our findings from chapter 3 are probably the most illuminating. We saw that canopy height responds really quickly. So these were month-long experiments. I didn't tell you that, but just a month-long, 4 weeks. And we saw really big changes in canopy height over that 4-week experiment. So we know that herbivores are a really strong control and a rapid control on turf algae. We have some data from an herbivore protected area in Maui that Emily and Levi are studying, looking at how herbivores structure the reef community. So I think that, you know, if we think of turfs as weeds as bad, and we know that herbivores eat them, like that's a really nice connection to protect herbivores, increase herbivores, and sort of decrease turf algae. I think the other thing is just educating reef monitors about collecting data on turfs. So it's pretty standard in coral reefs around the world, you know, marine reserves and managed areas for monitors to go out and collect fish surveys and reef surveys where they count the number of fish and they count the corals. And I think it's a really easy thing to do to also count turf algae and just be able to differentiate between like those sparse turfs that have a lot of bare space and are going to be okay for coral settlement, and those really thick, matted, dense turfs. Did I answer your question? Sure. You talk more later. Yes.
So one hypothesis that you gave for this kind of counterintuitive effect of when you have more fish, you see less of an influence of caging on turf.
In the invertebrates?
No, in the fish, in the marine invertebrates, yeah. So the hypothesis you gave was that there was more recruitment on, turf had already established in the places where there weren't as many fish, so they could grow faster. Is there a way to quantify like the number of recruits or the number of individuals or whatever to look at if that's in fact going on? Because it seems like you just look at the turf community and say, okay, well, we have a lot of recruits or established individuals on these reefs and on these turfs in Curacao.
Turf recruits?
Yeah, or how, I'm not sure how that would work.
Yeah, so the starting communities had more turf. The starting communities in Curacao had more turf than the starting communities in Millennium. So that part we do know. We didn't measure turf recruitment. It's a little bit tricky to measure.
You wouldn't necessarily want to get recruitment but to see if you have more growth potential.
Yeah. How is that different from just looking at like density, like how much turf you have to start with?
I don't know.
So what we don't have from Millennium is data on the turf plates before we put them into the experiment. And we do have that on Curacao. So that's one thing we can look at is on a specific, an individual plate level, individual sample level, like did those ones grow more or did you get more filament sticking up? And that is a metric that we could do. And we have those samples in the lab. If you wanna what you were getting at?
You brought it from it.
Yeah, it'd be cool to see if that process is actually happening, and I have to think a little bit about how to do that.
Right.
Yes.
So I like how you described the effects of consumers and resources as context dependent.
Thank you.
And you mentioned how you didn't think that your study sites were going to be at the extremes of nutrient levels or herbivore abundance. So I wonder if you could comment on, as far as like global management of corals, where like the sites, fall in this spectrum in that space of total nutrient level.
Yeah, how these sites compare to other sites?
Yeah, exactly.
Yeah, happy to. So those sites are all actually pretty healthy, relatively healthy. Millennium and Palmyra are off the charts in terms of fish biomass. They're some of the highest, those reefs have some of the highest fish biomass anywhere in the world. Curacao has a much lower herbivore biomass, but for the Caribbean, it's one of the best. So all three of those locations are definitely at the high end of the scale for fish. Curacao is sort of right smack dab in the middle of the range of what we'd expect for nutrient levels on reefs, and Millennium again is off the charts on the extreme scale. So what I said wasn't totally true. Millennium was extreme. Millennium is an extreme example. Curacao is a really average example. And so it might be cool to go to a place that has like no herbivores, which I know is extreme, and test that out. Yes.
I was wondering how fleshing out or algae fit into the picture. Would you say from your services, your services, that you think fleshing out algae are going to be your certain competitors with algae or corals? Do you see fleshing out algae increasing in abundance in the future more than turfs? Did you see any impacts from your fertilization experiments on macroalgal abundance?
I didn't measure fleshy macroalgal abundance in my studies, so I don't know about that. A lot of macroalgae species start life in the turf assemblage, and so many of the turf taxa that we looked at are just juveniles of what's going to become macroalgae. So there's this idea that if you let turf go unchecked for long enough, it will grow into a macroalgae community. The way they interact with corals is different. So macroalgae can shade over the top of a coral. Their fronds can abrade when they sort of move in the current. Turfs don't do that, but turfs do like collect sediment, which then has bacteria and anoxia and sort of is bad at the really small scale. They're sort of two different beasts. Generally, where you have stronger herbivory, you won't see as much macroalgae, and so there's sort of this spectrum of macro to turf to CCA. I think a lot of these global meta-analyses of how much algae is there on reefs lumps all of those things together. So when people talk about macroalgae, they're sort of sticking turf algae with it. I think turf is more common on reefs than macroalgae in general. 39.7%.
Okay, well, let's thank Jill again for her. I'd like to invite the committee. We're going to actually meet with Jill in room 4135 on the fourth floor pubs, and hopefully we will reconvene in here in about an hour with family and friends and colleagues. So thank you again for coming, and hopefully we'll see you soon. Like.
Did you have
Wait, are you everybody's screaming?